Hello students, welcome back to my biology online classes. After a pretty long time back, I am here with you to discuss about general characteristics of amoeba. Amoeba proteus is a typical example of subkingdom Protozoa under phylum Sarcomastigophora and subphylum Sarcodina. In 1775, R.V. Rosenhoff first discovered first time, and from the time of its discovery it is drawing attention of scientists because for its ever-changing shape. It is a single cell. Its body of amoeba is comprised of a single cell, and this single cell performs all the biological activities of its body. It is microscopic and body composed of minute, transparent, asymmetrical speak of protoplasm. Structure of amoeba is so simple. It is very easy to handle. So this amoeba is used in all level, from school level to university level, in all level, because of its simplicity and easily culturable. We are going to discuss about amoeba proteus. Why we are choosing amoeba proteus? Because it is simple most animal and its ever changing shape, and this name proteus it is taken from the fact of ever changing shape of Greek god Proteus. Amoeba means change, and Proteus, this belongs to Greek god for its ever changing shape. It is found in the bottom mud reach of organic material found in freshwater ponds, lakes, ditches, etc. under surface of the leaves of aquatic vegetation. And those who are interested or want to study amoeba, want to perform some practical activities, experiment about amoeba, how can they collect it and how can they make a culture of it? First of all, take some bottom mud rich from freshwater ponds, then keep it in a beaker, 200 cc beaker, and add some wheat grain in it, and after some days you will find an amoeba will appear in this culture, and collect the amoeba and see it under microscope. Obviously see it under microscope as it is a microscopic animal. Now let's discuss about general structure of amoeba proteus. As all of you know, amoeba is a microscopic animal, so the first point, it is microscopic. First point, it is microscopic. It is microscopic. Second point, microscopic, and it is single celled animal. Point number 3, 250 mm to 600 mm in diameter. Point number 4, irregular translucent mass of living animal like jelly. This jelly, it always changes its shape by sending and withdrawing its finger-like, and these finger-like projections is known as pseudopodia. Pseudopodia. Pseudo means not real. Podia means, pod means related to feet. Pod means related to feet, not real feet, so it's called pseudopodia. Single cell, this single cell animal, it is bounded all over by a delicate layer which is called plasmalemma. Plasmalemma, and below this plasmalemma there is ectoplasm, and below this endoplasm is surrounded by, this ectoplasm. Let's draw a cute diagram of amoeba proteus. This is a rough diagram of amoeba proteus. It has not a fixed shape. It is always ever changing in shape. So this is a rough diagram of amoeba, and this outer layer is known as plasma lemma, and its inner side this one is known as ectoplasm, and this inner layer it is known as endoplasm. This one is known as contractile vacuole. There is also will be presence of a nucleus. All of these are pseudopodia. These are minute. These are all food vacuoles. These are extending pseudopodia, and these are retracting pseudopodia, and the entire body dotted protoplasm layer. So this is amoeba proteus. Okay, this is clear. This outer layer, black colored outer layer is plasma lemma layer. Inner layer, this layer. It is ectoplasm layer, and endoplasm and inner endoplasm layer. This is surrounded by ectoplasm, contractile vacuole. This plays all activities of body. These are all about food vacuoles. This is nucleus, and these are pseudopodia, extending pseudopodia, and these are retracting pseudopodia. Okay. So if we want to study this diagram, the first point is plasma lemma layer. So let's discuss about some description of plasma lemma. Plasma lemma is a thin, delicate membrane. It is the outer living membrane of amoeboid cell. Plasma lemma, this is outer living membrane of amoeboid cell. Membrane is very thin, delicate and invisible layer of lipid and protein molecules. Its thickness is about 1/4 micron to 2 micron. If we see it under electron microscope, plasma membrane is composed of dark stained lines separated by a clear line. Some filaments are 80-degree angstrom in diameter extend from the plasma membrane to the medium, and outer layer of plasma membrane contain mucoprotein. The plasma membrane is selectively permeable membrane. Selectively permeable membrane means not all things passed through this membrane, only selective things can pass through this membrane, and it regulates the exchange of carbon dioxide and oxygen in between the animal and the surrounding medium. It also protects the inner protoplasm, and various ridge-like projections from the plasma membrane adhere the animal with the substratum. So it's all about the plasma layer. Then comes to inner cytoplasm layer. And the cytoplasm is differentiated into two layers, ectoplasm and endoplasm. The ectoplasm is outer hyaline ectoplasm and inner granular endoplasm containing various organelles. Next point is ectoplasm. It is thin, non-granular, hyaline and ridged and lies below plasmalemma layer and is considered as supporting layer. Ectoplasm is considered as supporting layer. Lanes from the cell to body. At the tip of pseudopodium, it forms a hyaline cup. Come to the endoplasm layer. This layer, endoplasm, innermost layer, endoplasm. Endoplasm is granular appearance. This is a heterogeneous mass. Heterogeneous means the density is not equal in all over the body. Heterogeneous mass surrounded by the ectoplasm. Ectoplasm is differentiated into outer plasma gel and inner plasma sol. And this sol and gel, this is the main key points of the locomotion of amoeba, which I will discuss later in my next classes. Plasma gel is relatively stiff, having granules which shows no movement, whereas plasma sol is a fluid, highly granular. It shows streaming movement. Beside granules, the endoplasm also contains organelles like nucleus, mitochondria, Golgi bodies, various different types, vacuoles of different types like food vacuole, contractile vacuole, etc. Also contains bipyramidal crystals, fat globules, and other inclusions. So this is all about endoplasm. Now comes to cell organelles. The first type of cell organelles we will going to discuss that. This is first point, plasmalemma, second point, ectoplasm, third point, endoplasm, okay, and the fourth one about cell organelles, and fourth one is nucleus. Let's discuss about some facts about nucleus. Amoeba proteus, there is a massive nucleus. Nucleus contains universally scattered chromatin granules. The nucleoplasm in small quantity. Under electron microscope, the nucleus is seen to bound of double nuclear membrane, having nuclear pore. There is honeycomb lattice. Under double layered nuclear envelope. The position of the nucleus is not fixed here. And this nucleus, this one moves with the shape of cytoplasm, move with the streaming cytoplasm. The nucleus plays a very important role in metabolic activity of amoeba, and chromosome number of amoeba proteus is 5 to scientists shape. Then comes to next organelle, my next and most important organelle, mitochondria. There are numerous rod-shaped and spherical. Each mitochondria has an outer and inner membrane. Cristae develop from inner membrane and project into the stroma. Stroma means the matrix of mitochondria. Cristae of rod-like mitochondria are many and very often it is branched. As usual, mitochondria supplies the energy source required for the cell activity. Golgi bodies. There are numerous Golgi bodies scattered in the endoplasm of amoeba proteus. Each Golgi complex is made up of 5 to 9 cisternae. It gives rise to primary lysosome containing hydrolase enzyme. Here Golgi acts as traffic police, guides the synthesized protein in different parts of the cell. The most important contractile vacuoles. Contractile vacuoles. So this one is contractile vacuole. Contractile vacuole. At the posterior end of the endoplasm, there is a round and pulsating. There is a round and pulsating vacuole. Round and pulsating vacuole filled with watery substance. And this water-filled round space is known as contractile vacuole. It is formed by the fusion of several small vesicles. As the contractile vacuoles continues to grow, it migrates towards the periphery. When it is fully grown, it discharges its contents by bursting. Into the water. Contractile vacuole is surrounded by several mitochondria. And this mitochondria, what is the function of mitochondria? It supplies the required energy for this activity. Mitochondria will supply energy for the functioning of contractile vacuole. The contractile vacuole, what is the function of contractile vacuoles? Contractile vacuoles serve the functions of osmoregulation and excretion. It removes CO2, it removes carbon dioxide. Carbon dioxide means CO2 or other waste from the body. The endoplasm also contains organic substances in the form of reserve food like droplets, fat droplets, bipyramidal crystals, and other important physiological functions: locomotion, nutrition, reproduction.